So, what do I do research in, and what do I think are some of the next stages in the treatment of epilepsy? So, one thing that you may have picked up on is I've been concentrating on how when anti-epileptic drugs act, they act on all the neurons without distinguishing which type they are in general. And in addition, with the deep brain stimulation or responsive neurostimulation stuff, where I showed the electrodes, that's also a technique that affects all the neurons that are in the area that's being stimulated indiscriminately. All right, so these are affecting large parts of the brain without targeting specific types of neurons. So my research is based on how can we affect a specific subset of neurons that might be even more effective at stopping seizures, right? So you may or may not know that there are lots of different types of neurons, different types of brain cells. There's some neurons that are excitatory, right? That may, when they fire their action potential, they may be telling the other neurons downstream of them to fire more, right? And there are also neurons that are inhibitory, where when they fire on their target neurons, they're telling those neurons downstream of them to fire less, right? And the way we get all the complex signaling in the brain is by kind of a balance of this excitation and inhibition and how it interplays. That's the complexity of the brain, right? So we have a lot of different tools in science to be able to target specific types of neurons. I won't go into it in too much detail, but what we have the ability to do is to insert genes into specific types of neurons, whether they're excitatory or inhibitory, such that that group of neurons that we've inserted this new gene into will respond to us, specifically to light. It's something called optogenetics, such that when you shine a light on the neurons that are expressing the gene you just put in, just those neurons that are expressing your gene will change their firing pattern. They'll either fire more or fire less, right? That's the basis of optogenetics, okay? And so since all neurons don't respond to light, if you had like an LED fiber optic cable or something that was shining light into a specific part in the brain, then if you turned that light on, most of the neurons wouldn't care. But if you had a gene expressing just, let's say, in these inhibitory neurons, and you shine that light, the only neurons that would respond to that intervention would be those neurons that you want to respond, right? And so I wrote a paper that came out in Nature Neuroscience recently that was using optogenetics in slices of human brain tissue that were resected from folks with epilepsy, that was donated to research, and showing that we can use these tools in human brain tissue. Now, the general way that this might work is that you can either choose to affect the inhibitory neurons and say, oh, when I shine my light, I want the inhibitory neurons to fire a lot more, and then tamp down the activity where the seizure is starting from. So you could turn on the inhibition. The other way around is if you just inserted your optogenetic gene into excitatory neurons and then turned on your light, if the optogenetic gene is an off switch, a sort of a don't fire switch, then when you turned on your light, those excitatory neurons that you have transduced with your optogenetic gene would then stop firing, right? And that could also possibly stop seizure activity. So the basis of my research is learning how to target specific types of neurons and to very, in a more focused manner, say, which neurons can we target in order to stop seizures. And so that's in broad strokes the kind of research that I do as a neurosurgeon and neuroscientist.